It's very complementary to the classical biology structure-based techniques. So I actually collaborate with a lot of people who do cryo-EM, cryo-ET, NMR, X-ray crystallography, but I don't do it myself. So a little bit of a different talk than the others that are in this section. But today I'm going to be focused on one of my projects that is a collaboration with Dr. Bin Yang, who's here today at WSU. And this is focused on demystifying the molecular mechanisms of glycoside hydrolases by glycoproteomics. So we already heard a little bit about this project with Brian earlier in the morning. So some of these slides might look a little familiar, but I'm going to go through them anyway. And so basically, we're focused on studying these fungal cellulases. There are several different classes of fungal cellulases. And really the focus of this work is to be able to implement these for biofuel industry. Because basically these cellulases, they degrade cellulose. And so this is a renewable energy source that we can hopefully utilize. The challenge with implementing this in large scale industrial applications is that these enzymes actually, their activity and the rate of hydrolysis actually decreases over time. And so we don't really know the exact mechanism of action of how these cellulases act on the cellulose, how they degrade the cellulose is not fully understood. And then also why that reaction rate is decreasing over time is also not fully understood. So that's really our main focus here, is to better understand these enzymes, and so that we can help to improve that overall reaction rate for industrial application in the future. Okay. So there's actually several different cellulases. They can chew up cellulose from different ends. We have two different major types of exoglucanases, which chew up on either the non-reducing or reducing end of cellulose. And then there's also endoglucanases that chew up within the center of the cellulose strand. And it's really the combination of these enzymes that improves the overall reaction rate. And so ultimately, eventually, we should study all of these so we can better understand all of these different cellulases and implement a combination of them into the industrial application field. The majority of studies that focus on cellulases currently, they focus on this particular cellulase which is part of the glycoside hydrolase family 7 class, which is called CBH1. It's from fungus Trichoderma rhizae, and it's highly expressed, and so it has been one of the major cellulases that has been studied for these applications. There's several different components of its structure. There's a catalytic domain, which is located on the right-hand side here, which we refer to as CD. And so that's where the hydrolysis actually takes place, where the cellulose strand is thought to be fed into this catalytic domain, and it hydrolyzes the cellulose strand into smaller fragments, which are basically, we call it cellobiose. It's just two glucose covalently linked to one another. There's a linker that's fairly non-structured, and that connects to the carbohydrate binding module, or CBM, which is thought to help bind to the cellulose to help with the affinity for cellulose. And also it's thought that it might have something to do with improving the activity of the enzyme. Previous research has also identified several different post-translational modification sites on this one. So it's not just this simple structure itself. There's also that added post-translational modification, and those there's several different N-glycan sites and O-glycan sites that have been identified. Those occur in all different portions of the protein structure, the catalytic domain, linker, and CBM. And so some previous MD simulations have hypothesized that these glycosylation affects the activity or the stability of the structure or of the enzyme. And so previous work from other researchers, they aimed to identify what each of these different glycans, how mutating those site-specific sequences within the structure, how that affects the thermal stability, proteolytic stability, as well as the enzyme activity. And so when they're looking at specifically mutating CBH1 at the positions for where there's N-glycan, where it can be post-translationally modified to have N-glycans, in the catalytic domain, they found that it's important for thermal and proteolytic stability. O-glycans are in the carbohydrate binding module are not involved in stability or activity, but they do aid in enzyme folding. And then the O-glycans in the linker, they dramatically affect proteolytic stability, and they also perform some MD simulations that suggest involvement in binding affinity to cellulose. And so these studies really only capture an all or nothing approach, where they're either completely removing that ability for the glycosylation to take place at particular sites within the amino acid sequence, or they're comparing it to the overall distribution of glycans that can take place at that particular residue. And so really understanding how the difference in post-translational modification at each particular site, if there's some variability in that post-translational modification, that's also an aspect that we hope to tackle with our work. This is based on MD simulations, so this is just basically a hypothesis, a guess of how CBH1 performs its activity, where it binds to cellulose. There's a recognition step, threading of the cellulose strand into the catalytic domain. There's complex formation, hydrolysis of the cellulose strand into cellobiose, and release of that cellobiose. There's also a lot of other MD simulations. And they also include in their structure the known sites for post-translational modification. But again, these are just guesses. They're not, there's no experimental evidence to support this, this hypothesis. Again, another, you get the idea. There's a lot of different models and simulations for what might be going on with this enzyme. And so really tackling this challenge can be, it's very challenging with the classic techniques for structural biology. And so what we want to do is implement several complementary techniques to your classic structural biology techniques using mass spectrometry. So we have several different techniques that we're implementing here to help address some of these questions that we have on CBH1's activity, what affects its activity, and its structure. And so we take, this is CBH1 here in the center, just a simplified model. We can take that, and a lot of people I think here are familiar with bottom-up proteomics, where you're basically taking your protein, digesting it with your favorite enzyme, your favorite protease, and this is, then you inject this onto the mass spectrometer, these small peptides, and this is really great to identify precise sites for post-translational modification, because you get really good sequence coverage using bottom-up proteomics. However, although you can identify these particular sites, you don't really get very much knowledge, if any, on the combination of these post-translational modifications on the intact protein itself. And so that's what top-down proteomics is doing here. So rather than chewing up that protein, digesting it with a protease, we look at the intact protein within the mass spectrometer, so we can get information on intact proteoforms. And so this way, now we can start parsing what are the different potential intact proteoforms that are present in our sample with those different glycans that we identified for CBH1. And then we're also combining this with native mass spectrometry. And what's really cool about this technique is that we're able to maintain non-covalent interactions in the gas phase of the mass spectrometer. And so we can study protein-protein interactions, protein-ligand interactions. In this case, we're looking at substrate and product binding, and we can also track reaction progress in just a few microliters of sample. So although this doesn't really replace the classic structural biology techniques, it is very complementary and it can aid a lot of your work. So if you're interested in collaborating, let me know. All right, so the first part of my talk, I want to focus on basically combining these techniques. We want to validate that we can do this for the system. So we're looking at CBH1 wild type. We also have a CBH1 mutant, catalytic mutant. So that's the E212Q mutant here. So this abolishes the activity of the enzyme. And then we're also looking at the catalytic domain by itself. And so, yeah. So I'm just going to summarize the bottom-up proteomics data in this one slide. But basically, this is the sequence of CBH1. The yellow highlighted region is the catalytic domain. We found several different N and O glycan sites within the catalytic domain sequence. This matches to previous literature results. This portion that's not highlighted, that's all red here, is the linker region. We also identified several O glycan sites there. And then in this kind of orange highlighted portion at the end, that's the carbohydrate binding module. We identified several O glycan sites there as well. And then we also looked at both the wild type as well as the mutant, the E212Q mutant, and the catalytic domain by itself. And we see similar post-translational modifications that we did for the wild type. So now what does the intact proteoform distribution look like? So now we're looking at top-down proteomics data. And so basically you're looking at the mass on the x-axis, and we can see each of these peaks is a different proteoform. And so with the wild type CBH1 sample, we see peaks that differ by 1 hexose unit, so 162 Daltons between each of these peaks. And so this really demonstrates how it's not just a simple system where we have a singular proteoform in our sample. We have a whole distribution of different proteoforms for even our CBH1 wild type. Then when we look at the E212Q mutant, we see a similar pattern where we have these peaks differ by 1 hexose unit. However, the proteoforms, intact proteoform distribution is different from the wild type sample, and really highlights the value of using top-down proteomics as to assess whether or not you have any changes in post-translational modifications between your samples. So imagine if you're comparing between wild type and the mutant, and just assuming that it's just a single amino acid substitution. Well, that's maybe not necessarily the case. So this is a great method to be able to analyze whether or not you have any changes in the intact proteoforms that are present in your sample. We're also looking at the catalytic domain, which you'll see there's not a lot of different proteoforms that we're seeing, mostly just one single proteoform present in this sample, and this matches previous literature. And so it's kind of what we were expecting to see. So this really highly suggests that most of this heterogeneity that we're seeing in the wild type and the E212Q mutant is coming from the linker region of the protein or in the carbohydrate binding module. So let's look at some native mass spec data. So this is a lot, I know. I'm going to go through it. Yeah, here we go. Okay, so this is the wild type. These are the intact proteoforms. This might look familiar from the previous slide. And so this is the most abundant intact proteoform that I've highlighted in red. And so we can use that to then track the binding of our different soluble substrates. We have 2 soluble substrates, cellotriose and cellotetrose. And so cellotriose has 3 glucose. And so we'll see a shift to 3 here, which is labeled. So that's indicating binding of cellotriose to the wild type. And then cellotetrose, we actually don't really see binding. The enzyme activity is so fast that we're actually seeing cellobiose, the product formation, which is the 2 here. bound species. We can also look at the E212Q mutant. We see binding without reaction, which is what we would expect with a mutant that abolishes activity. And so we see binding of cellotriose with the 3, and we see a shift to the proteoforms with 4, 4 here meaning cellotetrose bound to this most abundant intact form. Yeah. The catalytic domain also has cleaving with weak binding, so we can also track the binding of cellotriose and production of cellobiose. And similarly, cellotetrose, we see actually cellotriose and cellobiose binding. All right, the next part of my talk, I want to, so we wanted to delve more into detail on tracking the reaction progress and structural changes of CBH1 wild type. And we're also looking at another cellulase, an endoglucanase, AG1, in combination to see how that affects the reaction progress. And so to do this, we're doing native mass spectrometry. So we're looking at soluble and insoluble substrate binding. We're tracking that binding and reaction progress over time. And then we're detecting changes in protein structure, combining native mass spectrometry with ion mobility. So with CBH1 binding to cellotriose at 5 minutes, we can see this blue circle is denoted for CBH1 unbound, so the apo species. We can see 1 bound cellotriose, 2 and up to 3 in the 5-minute sample. And then after 18 hours, we still see bound cellotriose, but we also see the increased abundance of the species associated with CBH1 bound to its product, cellobiose. We can also do this with the other soluble substrate, cellotetrose. We don't really see cellotetrose bound, but we do see, because we think the reaction is quite fast. So we see cellotriose bound and cellobiose, so different distributions here. We're also looking at CBH1 binding to an insoluble substrate, and we collaborate with Brian, who spoke earlier. We can't really track in native mass measurements the insoluble portion of the sample, so Brian does that with his measurements, but we can track the binding of product over time because that will be soluble. And so at 5 minutes, we only see the apo peak for CBH1, and after 18 hours, we see the emergence of this cellobiose bound CBH1. What other information can we gather from these experiments? Well, if we're looking at the bulk solution substrate and product here on the left, we can actually track reaction progress within the same few microliters of sample. And so that's what we're looking at in the low M/Z range here. And we can also isolate the protein species here, CBH1, on the right. And we isolate that in the mass spectrometer, add a little bit of collision energy, and we can actually release bound substrate and product and confirm that that is in fact what we are seeing bound to our enzyme. So it's kind of a confirmation. So this is a summary of the binding and reaction progress. It's, I don't know if I have too much time to go into it, but basically, we did these experiments with CBH1, EG1 by itself, CBH1 and EG1 together. We can track the binding here. So we see a lot of binding of CBH1 to cellotriose, and a little bit of the product bound species. I do want to highlight EG1, where we don't see a lot of binding to substrate and product, but if you'll note the cellotetrose, we see a lot of reaction progress for the cellotetrose sample, where primarily a lot of the cellotetrose has been converted to cellobiose after 18 hours. So we can't really, we can see a little bit of bindings of the this should say cellotetrose, cellotetrose to EG1. It's a little bit difficult to resolve, but we can also use that bulk solution substrate product, and we can detect that formation of cellobiose, so we can track that reaction progress. And then we can also detect the release bound cellobios by adding some energy. The other thing I wanted to highlight is the apo species of CBH1. And so you'll see at 5 minutes and then 18 hours, we have quite an increase in the apo species after 18 hours, but we don't really see a lot of reaction progress. Most of the triose or cellotetrose that's in the sample has not been converted to product. And so yeah, we want to, so this gets to the question of what is causing this, what is causing this? I think I have not a lot of time, but basically what we want to look at is maybe if there's any change in the proteoforms, intact proteoforms in our samples. And since I don't have a lot of time, I will basically just say the short answer is no, we don't. And so compared to the control, we don't really see any obvious changes in the intact proteoforms. We do in the future want to see if we see any differences in the individual sites that are post-translationally modified. So that is a future direction. And the last thing I want to highlight is then also adding ion mobility to our measurements so that we can study protein structure. And so basically we have this drift tube within the mass spectrometer. We have an electric field applied, and essentially we have an inert gas in this tube, and that, depending on the size, shape, and charge of the species, of the protein species, we're able to separate larger species drift slower through the drift tube because they're bombarding with that inert gas more, and so we can basically separate based on conformational changes of the protein as well as different size protein. And so some preliminary data here is just showing examples of this where we're taking those intact proteoforms and then we can extract the drift time data and compare between the controls versus the substrate added species. And so we see some potential, at least moderate shifts in the drift time. And so this is something that we're planning to explore more in the future. And so I guess basically my summary, I'm not going to read all of this. That's a lot for me to read, but basically I hope that today you have learned at least a little bit about native mass spectrometry and what this technique can do for you to help complement some of your structural biology techniques. And so with that, I guess I will open the floor to questions.
Thank you, Stephanie. We're running about 10 minutes behind, so no, not your fault, it's related. If you have any question for Stephanie, please reach out to her after this session is finished. Thank you very much.